You found the skeleton. How would you calculate the depth? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hey there, YouTube, the Dapper Dinosaur here. It's time for a long-delayed update to the Speculative Evolution project. This time we have 100 new organisms, but only if you count in binary. Anyway, I do want to apologize for not getting this out sooner, but computer problems have kind of thrown my mojo off. However, at this point, I think I'm more or less back on track, so hopefully it won't be another month or more again for the next video. One more thing before we begin, if you could please remember to hit the like button and subscribe if you aren't already subscribed. It really helps me out. The YouTube algorithm is brutal, and every like and subscription really helps get these videos out there, and the more they get seen, the easier it is for me to do these videos. But that's enough preamble. Let's dive into some cool new organisms made by you, the fans. First up, we have the horned snail. Amongst the surviving cephaloptins within Almaish's ocean some 440 million years ago, there exists two principal groups. The first of which is Chiliphoichthyes, a rapidly diversifying group consisting of those cephaloptins that have developed a hardened carapace from their notochord-like stems, which evolved from some of those early cephaloptins, which remained principally benthic organisms. And Esotericae, those cephaloptins whose ancestors took to the water columns as a means of escaping predation, is the other, to which the sea sweepers and rock eater belong. This early division would be expected to result in clean division between the organisms. The fact that such division involves such clearly recognizable characteristics only serves to the benefit of paleontologists, ecologists, and xenobiologists, who have all come to work in the study of Almaish's ancient biosphere. However, such clean division between clades to such degree as to suit the human mind is often not the case in nature, and such has proven true in the xenosuperclade Cephaloptina. The division between Esotericeans and Chilioichthyans is, while incredibly ancient to the superclade, not an immediate division. Fossil evidence of the earliest cephaloptins show traits of both clades comparable to the extinct Guionieros, and its relationship to the ray-finned and lobe-finned fish of Earth. Seldom are two coins alike, and this is also true for Cephaloptina and the bony fish. For instance, unlike the bony fish, the ancestral cephaloptin is believed to have been discovered, nicknamed the pudgeworm, which was once believed to solely be the ancestral Thalassalicarian, though this is not currently held to be true. This discovery has proven crucial to understanding the evolutionary history of cephaloptins and their relation to other xenosegmentans, as well as phylogeny within the superclade itself. However, for all of these insights, a particularly glaring problem with phylogeny regarding the early cephaloptins has been brought to the forefront. This is where the clade Thalassolincari enters the picture. The Thalassolincarians, meaning sea snails, are a clade of basal cephaloptins closely resembling the ancestral pudgeworm, and show a particular combination of traits that prevent them from being classified as either members of Esotericae or Chilioichthyes. The specific definition of this term was originally identified as the most recent common ancestor of the bar snail, Losos malachio, crowbar mollusk, and the quadrihorn, Thalassierptoloselincari, the snail crawler snail. However, with the discovery of the pudgeworm and additional analysis, it has been decisively proven that the pudgeworm is karyotypic of the last common ancestor. Thus, this group would technically include all cephaloptins, including the clades Esotericae and Chilioichthyes. Thus, this group is now understood to be paraphyletic, and is now defined as all cephaloptins who do not belong to either Esotericae or Chilioichthyes. On Almaisha some 144 million years ago, all cephaloptins belonged to this group, and were fairly basal compared to the Chilioichthyan and Esoterican kin, and most closely resembled some alien form of shellless sea snail. Within the clade are several orders and families, including members more closely related to Esotericae or to Chilioichthyes than other members, as well as some not closely related to either, at least as far as cephaloptins go. On the Chilioichthyan end of Thalassolincari rests an order of organisms referred to as the Keratobrastrae, or horn arms, within which a variety of organisms called horn snails resides. These organisms, while being closely related to the ancestral Chilioichthyan, lack shell development of any form, a trait that the ancestral Chilioichthyan possessed. Pictured, an artihorn snail wanders looking for food, creature designed by Lethal Cuteness and Dr. Misa. The horn snails are small, soft-bodied herbivorous organisms with stocky, pudgy bodies and small heads. Segmentation on their bodies is still present, a leftover trait from their Xenoeuli ancestor, and non-functional opsin clusters remain present. The number of segments have been significantly reduced, however, as segments 3, 4, and 5 have completely merged into one tagma, with the first 4 limbs. Segments 6 and 7 have also merged and now consist of the back 2 limb pairs. Segments 8, 9, and 10 are merged in the original cephaloptin ancestor. However, in the horned snails, this tagma has shrunk significantly and now serves simply as the container of the posterior anus and gonads. The organisms possess 4 bony fins, each located on the underside of the body. These fins serve as walking limbs and have the ability to grasp the sediment due to the internal anatomy. Within each fin rests a set of several bones, each with several muscles between them. The bony structure itself is anchored to the notochord through a long muscular hydrostatic structure. These anatomical features exist in at least some form within all cephaloptins, and within the horned snails, they are used to create a walking structure. These limbs help to serve anchor the horned snail to the sediment, and when combined with muscular hydrostatic structures lining the interior of the body and connecting them to the notochord, allow the organism to walk along the substrate. These limbs serve the additional purpose of allowing the horned snail to dig into the sediment if danger approaches. Due to its general lack of bones and soft body, the horned snail possesses the ability to squeeze itself into openings far smaller than itself. Given the organism's already small initial size, the horned snail is capable of squeezing into spaces as small as 10 millimeters. Within the horned snail's body tagma exists an esophagus on the roof of which exists a small pouch-like ridge that contains 2 ctenidia. These ctenidia serve as the primary respiratory organism for the creature, as its gill fronds, while still functional, are located in a highly unfavorable position for acquiring oxygen from the surrounding water. Thus, these gills are used principally as a means of removing carbon dioxide and other aqueous waste products from the bloodstream, while the ctenidia are used for both acquiring oxygen and expelling carbon dioxide. The esophagus connects to a hatch-protected stomach, which can open to allow food and particulates to enter the organism's system. This stomach is connected to a gland system, which produces stomach acids and enzymes to digest food. This in turn connects to a long, winding intestinal tract that runs from the stomach to the posterior anus. As this organism does not contain a bladder to hold waste products, they are expelled the moment the posterior anus is opened. It may be sealed with a small muscular hydrostat surrounding the rear of the intestinal tract shortly before the opening. Within the mouth of the organism rests a radula covered in small keratin teeth, anchored on an odontophore located on the roof of the mouth. This radula can be pushed downwards to effectively seal the mouth, and is used to ensure that edible material is ground into a fine particulate as it is scraped off the sediment and sent through the mouth and into the esophagus. Behind this is a pair of osphraphidia, which are used to gain information regarding the type of particulate entering the organism's throat. This allows it to roughly gauge if the particulate it is consuming could be harmful to its ctenidia, and thus helps ensure that the water it is intaking does not possess harmful particulates. This organ function can double as a means of ensuring that the food particulate is ground up enough for efficient digestion. However, this process, governed by instinctive behaviors, is incredibly basic and is insufficient to properly measure how well chewed its food is. The nervous system of this organism, like most other thalassolemcarians, is quite basic. It consists of a simple cluster of nerves in the upper portions of the head tagma, 2 bilateral nerves running alongside the notochord, and 2 neural rings connecting them with the body tagma. Connected to this are 3 pairs of statocysts, filled with a small amount of hemocyanin and air. These statocysts are utilized by the organism as a means of detecting gravity and are located at 3 strategic portions of the organism. 1 pair is in the cranial tagma, and 1 is between each pair of limbs. This allows the organism to detect how gravity is acting on its body, and thus allows it to position itself in favorable positions for feeding and retreating from predators. Perhaps the most surprising anatomical feature of the horned snails is their cranial tagma. Like all cephaloptins currently in existence, the horned snails possess 4 distinct camera-type eyes. However, in the horned snails, these eyes are located along long, muscular hydrostatic structures resembling the arms of cephalopods or the antennae of snails and slugs. Each eye is located on a separate tentacle that possesses a significant range of motion. This is the horned snail's main method of defense: detection. By placing eyes along long hydrostatic structures, the organism is able to maintain an extreme range of vision at all times and can effectively watch for danger both while feeding and hiding. Through the use of its camera-type eyes, the horned snail can detect danger from a significant distance. Though it lacks stereopsis, the simple ability to see danger is enough for the creature to begin burrowing into the sediment. As a hole does not need to be particularly large, the organism is capable of burrowing rather quickly or escaping into crevices that are impossible for predators to reach. It can then use its 4 eyes to peer out from its hiding location to see if it is safe to emerge. As an interesting side note, horned snails get their name from these muscular hydrostatic structures holding the eyes. These structures were originally misidentified as horns in the holotype due to their position and appearance, thus the name horned snails. However, it was later determined through analysis that these organs were in fact muscular hydrostatic structures, not horns. Horned snails are diploid hermaphroditic organisms. In the third tagma on either side of the notochord is a gonad. 1 gonad is male and 1 is female. When conditions are optimal, usually defined by the warm, nutrient-rich waters of the summer month, and assuming the horned snail is healthy, horned snails begin to release hormones and enzymes bound to their waste material into the water. These hormones are detected by the osphragida, which allow the horned snails to follow the scent. When the horned snail has located a mate, they will begin to dig a burrow or locate a suitable mating site nearby. Possible sites include crevices between reef structures and under rocks. When a proper nesting site is found or completed, the horned snails will each lay their eggs before fertilizing the others. The horned snails will guard the fertilized eggs for 1 week until they hatch, after which the horned snails will leave the area and feed for several days to regain strength. Afterwards, assuming conditions are still optimal, the organisms will attempt to mate again. The initial stage of life begins with eggs, which are laid in clusters of 20 to 50 in a suitable burrow. During gestation, the parental horned snails will attempt to protect and incubate the eggs to ensure that optimal environmental conditions are maintained for development. However, they will prioritize their own lives over their eggs and will still flee from predators if given the chance to do so. After 7 local days, the naiads will emerge as miniature versions of the adults. These small organisms are barely large enough to prevent themselves from being dragged off by the current and must cling to the substrate until they are large enough for their weight to anchor them down. During this time, they will feed exclusively on small algae-like retinophytes and phytozoans growing on rocks and corals. Some may even live among the structures of the fortress mesas, eating small plants growing along the structures. After about 7 local days, the young horned snails are large enough for their weight to anchor them to the surface they crawl on. It is also around this time that the young horned snails will begin to feed on small animals and occasionally scavenge carrion. For those that live near fortress mesa hives, a ready source of food in the form of scraps from scavenged material and leftover biomass on the shells. For others, algae-like phytozoans or retinophytes which supplement the carrion and microbes is sufficient. After an additional 14 local days, the horned snails reach their adult size. By this point, the gametes develop fully and sex hormones begin to be produced. These hormones will be released when conditions become favorable for reproduction. At this point, the organism is considered to have reached full maturity. The horned snails prefer habitats where there are plenty of structures for them to burrow into or crevices to use as hiding places. For this reason, they prefer the crowded shallow seas of the Yama-Kubshai reef system, where the crevices and food are abundant. As they lack any form of hardened carapace or external defensive structures in general, their main strategy to escape predation is to flee as opposed to fighting. Horned snails prefer to avoid confrontations when able, and exploit their feeble muscular bodies in doing so. When confronted with a predator, horned snails will quickly use their eyes to observe their surroundings. If able, they will flee into any tiny crevices between reef builders and rocks, and if unable, they will begin to burrow deep into the substrate. Their fins are bony, and the muscles controlling them are some of the strongest in the creature's body. As a result, the organism is able to burrow deep into the substrate fairly quickly. The organism will prefer to hide in these crevices until the danger has passed, after which it will leave and resume its normal behavior. Thalassiarpito salankari, the quadrihorn snail, this species lives around the tip of southern Yama, as well as all across the shallows of northwest and western Kubshai. T. hydra, the Yamahorn snail, this species lives around Yama on all but the northern coast. Keratomati tenome, the Artihorn snail, this species lives around the east and southeast coast of Arctica, and the east and northeast of Yama, and the northernmost parts of Kubshai. K. microa, the flathorn snail, these live all through the Yama-Kubshai major reef system, as well as smaller populations around the northeast coast of Yama. As a horned snail has practically no defenses if caught, all of its biological adaptations regarding escaping predation are dedicated to escaping via retreating into locations where predators cannot follow. This has proved quite effective against most predators. However, it has led to some predators specializing to hunt them specifically, as they are a fairly nutritious and reliable source of food for such a predator. The specific dietary needs between species vary, however the general diet remains the same. Horned snails are primarily herbivorous organisms feeding on small sprouts and algae-like retinophytes and phytozoans. Between the two, horned snails are significantly better at digesting phytozoans due to the composition of the enzymes produced in their digestive gland. The exception to this is K. microa, which prefers to digest retinophytes. This switch is in part due to the presence of chemicals in their digestive tract, as several byproducts of their reactions are useful in the synthesis of digestive enzymes or play a direct role in digestion. Rather than rely entirely on herbivory, horned snails also act as opportunistic scavengers when the opportunity arises. In addition to this, they will also digest xenometazoan microbes as well, though this plays a far less significant role in their diet. The reason they do this is to gain easier access to proteins and amino acids that are easier to digest than those in retinophytes and phytozoans. Their diet is still primarily herbivorous in nature, thus allowing them to avoid competition with scavenging organisms within the reef system. As another means of supplementing their diet and gathering means of aiding in digestion, horned snails will also consume debris and small pebbles in the substrate. These gastroliths serve 2 main purposes, digestion and symbiosis. Horned snails utilize gastroliths as a means of aiding in breaking up small clumps of plant matter, grinding them into even finer powder to further aid in digestion. Additionally, these gastroliths aid in the cultivation of pockets of chymocoids that produce compounds useful in digestion, enzyme production, and pH regulation. This chymocoid cultivation is not necessarily deliberate, the flathorn is an exception, but rather simply a beneficial side effect of the consumption of gastroliths and life on the seafloor. K. microa, unlike most horned snails, prefers to consume and digest retinal phytes as opposed to phytozoans, and they have adapted their digestive enzymes to do so. In addition to producing enzymes to attack the cell walls of retinal phyte autotrophs, the flatworms make use of a greater variety of digestive aids that are produced by chemicoid symbiotes living within their gut, which feast on the carbon dioxide produced by cellular respiration. These flatworms possess many blood vessels that diffuse carbon dioxide into the digestive gland, where most chemicoids reside within the creature. They then use enzymes and transporting hormones to secrete productive byproducts into the stomach and the bloodstream, while harmful byproducts are sent directly to the posterior anus. Due to an increase in the need of mineral compounds, the flatworm will consume a larger quantity of gastroliths to help grind up plant material. Most horn snails make use of some gastroliths to help further grind plant material. However, the flatworm regularly consumes debris to supply its chemicoid symbiotes with minerals to perform chemosynthesis and produce helpful byproducts that can be utilized by the organism. T. selinkari, the quadrihorn snail, is a species of horn snail descending from the western shores of Kubshai and spends most of its life in the shallowest regions of the reef system that are sufficiently distant from the murky river outlets. These organisms possess 4 long eye stalks that are relatively identical to each other. This type of horn snail is one of the most common on Almaisha and has a range extending from southwest Yama to northern Kubshai. T. selinkari, the Yama horn snail, is closely related to the quadrihorn snail and shares a portion of its range with it. However, unlike the quadrihorn snail, the Yama horn snail's range consists of the coastline of Yama, hence the name Yama horn. As the continent of Yama consists of more lowlands, estuaries, and wetlands, the Yama horn has adapted to cope with the waters that may be murkier. Its eye stalks have grown significantly longer than those of the quadrihorn to grant it greater visibility away from its body, and its ostrophidias have adapted to become somewhat of a scent-detecting organ. In this way, the Yama horn is able to smell organisms in a way that is somewhat analogous to tasting the water for them, thus helping it detect predators in the event its eyes become ineffective. Keratomati tenome, the artihorn snail, gets its name from its range. Much like the Yama horn, rather than inhabiting the shoreline of Yama, the artihorn lives around the southern coast of Arctica, with some small populations living in northern Yama and Kubshai, where competition with other horn snails is reduced. The horn snails, while visually more similar to the genus Thalassier are actually more closely related to the flathorn. In fact, fossil analysis indicates that these two organisms were so closely related that they were likely part of the same genus, Keratomati. The artihorn snail lives a similar lifestyle to other horn snails. However, it is better adapted to more extreme seasonal weather fluctuations in temperature and the day-night cycle. As such, it has succeeded in outcompeting encroaching populations of the genus Thalassier. Keratomati microa, the flathorn snail, is by far the most unusual horn snail, at least compared to other members of its family. Unlike most horn snails, its eye stalks are actually quite short and stubby. It is likely that this organism is closer to the ancestral horn snail in morphology, as there is no fossil evidence of a regression of eye stalk length from this period. While the flathorn snail lacks the large range of visibility that other horn snails possess, it does possess a more specialized digestive system, more specifically tailored to retinal phytes, thus avoiding competition. Its flat eyes, while unable to rotate for 360-degree vision, do provide the organism with exceptional front-oriented vision. Thus, while the species must turn its head in order to observe its surroundings, it is able to better analyze visual information. As a result, this species also demonstrates superior decision-making skills as opposed to its relatives. The horn snail, like all other cephaloptins, possesses a camera-type eye. This visual sense serves as the primary mechanism for protection against predators and environmental hazards, as it allows the organism to detect danger visually from great distances, oftentimes around 100 meters or more in ideal conditions. Additionally, neurons lining the underside of the limbs of the organism allow it to readily detect sediment conditions beneath it. These two senses allow the organism to maintain awareness of its surroundings constantly, thus helping it detect dangerous conditions before they can impact the creature's life. In addition to these sensory organs, the horn snail possesses 6 statocysts arranged in 3 pairs throughout its body. Each statocyst contains gases such as carbon dioxide expelled from the bloodstream and a small amount of hemolymph or water. Within this organ are thousands of tiny cilia which are used to detect the orientation of the liquid in reference to the gases contained inside. These organs allow the horn snail to know how its body is oriented relative to the direction of gravity, down, and thus aid it in positioning its body to maintain awareness of the organism's location in the environment. Lastly, the horn snails possess an ophrydia just behind its radula. This ophrydia is designed to function as a means of detecting particulate matter within the water column. This organ allows the horn snails to maintain a relative awareness of the water it is intaking into its stomach and exposing to its tenidia, thus helping it to ensure that it does not intake harmful sizes of particulate. This organ also serves other purposes for the horn snail. Notably, it can help the organism ensure that its food is ground sufficiently so that it may be easily digested, and it can also help the organism maintain awareness of the consistency of the water in other ways than sight. If the water is particularly murky, the organism can detect this both through visual indicators and through employing its ophrydia. Based on this information, the horned snail is able to act accordingly to protect its gills while still maintaining necessary gas exchange for its metabolism. Genetic ancestor, Xenoeuli kirbii. Scientific name, Flaciarpeto species and Keratomati species. Origin, Xenosegmenta. Lifespan, T. hydra 5 local years, T. salankari 7 local years, K. microa 5 local years, K. tenome 4 local years. Average height, T. hydra 4 cm, T. salankari 5.5 cm, K. microa 4.8 cm, Keratomati tenome 3.9 cm. Average length, T. hydra 8 cm, T. salankari 12 cm, K. microa 10 cm, K. tenome 8 cm. Next up is Lutolacerta. In the freshwater systems of Yama, Lutolacerta fills the niche of a medium-sized omnivore. It is the largest member of a group of bobird descendants who have developed a cartilaginous skeleton to support their internalized gills, referred to colloquially as gill cords. Lutolacerta is primarily a bottom dweller, using its large chemoreceptors to guide it through the murky waters for food. It will scavenge primarily, but is capable of active hunting due to its powerful tail. Pictured here, a pair of Lutolacerta forage around the edge of a Yaman lake. Creature design by Saurus Blooded. Like all gill cords, Lutolacerta has an internal cartilaginous skeleton to support an internal gill system. This protects the gills from parasites or being damaged. The tail has 8 pairs of rounded spiracles positioned laterally that allow for water to enter the gills. The tail also possesses a flattened membrane that is used for propulsion when Lutolacerta is actively swimming, though it usually spends time walking on 4 padded feet made for digging in muddy substrate. They are rather short and possess 2 toes each. The body is more domed and aerodynamic to allow for a much more active lifestyle. Lutolacerta is jawless like its ancestor, but possesses a sharpened radula for masticating food. Its eyes are larger than its ancestors. The chemoreceptors on the lateral sides of its head have become larger as well as wider at the tip. Lutolacerta are hermaphrodites who breed twice a year. When a receptive mate is located, they will begin digging a pair of burrows in muddy sides of a lake or pond. They will then both lay eggs, one in each burrow, and fertilize them before sealing the burrows. The pair will then defecate around the burrows as a way to mask the scent of eggs. After a period of 30 local days, the Lutolacerta eggs will hatch and emerge from the burrow in a larval form. The larval form resembles the ancestral bowbird except for the gills being internalized. They will live on the lake or pond bottom as detrivores over a period of 1 local years until they reach a height of 4 centimeters. At this point, their bodies will begin forming their adult features, the radula and large chemoreceptors and coloration. This process will take about 60 local days, after which they will be fertile adults. Lutolacerta is named for the freshwater rivers, lakes, swamps, and ponds of Yama, though it can only breed in slow-moving water with a muddy bottom. Lutolacerta are omnivores who feed on retinophytes, juveniles of other species, carcasses, and eggs. Lutolacerta's eyes have improved to be more sensitive to movement, but its chemoreceptors have become far more sensitive and able to detect buried food sources, like the eggs of many freshwater fauna local to Yama. Genetic ancestor, bowbird. Scientific name, Lutolacerta pictoratus. Origin, pseudotetrapoda. Lifespan, 8 local years. Average height, 7 centimeters. Average length, 31.5 centimeters. Now we come to the radio hedgehog. Despite the name, the radio hedgehog is not an 80s British pirate radio station. Instead, it is a sessile radiotroph that looks a bit like a cactus and a hedgehog and a sea urchin all combined. Radio hedgehogs are, in fact, descended from feather bushes, but unlike their ancestors, they forgo the nectonic part of their life cycle, instead growing into permanently sessile structures. Pictured here, a small stand of radio hedgehogs grow on a now cool lava flow off the coast of Nyland, where the water is still a bit too toxic for most organisms. Creature design by Dapper Dino. Radio hedgehogs have a relatively simple anatomy at the trunk and main cap. The tissue is light and full of simple cavities with seawater in them, but also there are tubules distributed throughout the organism that distribute nutrients. The interesting bit is the spines. Each spine forms a small bud on the top of the surface of the cap. Once it is large enough, it will start to extrude a hollow filament of copper with a sharp barbed tip. These filaments have a square cross-section and are only a few millimeters thick. These filaments are essentially the leaves and reproductive structures rolled into one. When full grown, a radio hedgehog will broadcast spawn isogamous haploid gametes on full moons. Very few radio hedgehogs grow from these fertilized spores that sometimes develop from this. As a result, radio hedgehogs do not actually broadcast nearly as many gametes as typical for a broadcast spawner, instead focusing most reproductive energy on their spines. Life for most radio hedgehogs starts as a spine bud that grows from the cap of an adult. When the spine on the end comes in contact with the soft parts of a nectonic organism of sufficient size, it may embed itself in a tissue. While this is painful, the wound is not deep or large, and if the organism avoids infection, it is unlikely to cause any long-term harm. Further, because the spine is primarily copper, which is antimicrobial, infection is not terribly common. After a few hours of swimming, the barb on the end of the spine will break from brittle fracture, or the organism may scrape the bud off of it, if it can. Either way, the now detached bud will start to grow into a new adult, provided there is enough material and radiation for it to do so. It will resorb the copper spine and use it to help build its body. Hedgehogs grow in the coastal waters around Nilan. While still primarily feeding on ionizing radiation from fresh volcanic activity, and using the minerals released by such activity for most of its body structure, the radio hedgehog has developed a unique form of photosynthesis. It uses the photoelectric effect to produce supplemental energy that is now available in its shallow habitat. Special tissues line the inside of the spine and act as wires to transfer energy in the form of electricity proximally down the bud, and then distribute it to the rest of the organism. Genetic ancestor? Featherbush. Scientific name? Pseudoerinaceus chalcophoros. Origin? Radiotropha. Lifespan? Until radiation flux is too weak, typically about 2 years. Average height? 25 centimeters. Now we come to Saphia ziwa. If someone collected a cup of water from the lakes and swamps of Yama about 440 million years ago, the most likely species they would find are the microscopic Saphia ziwa, or lake cleaners in English. These diminutive descendants of Xenoeule kirbii have adapted to be the basis of life in the freshwater ecosystems of Yama. They are responsible for the majority of waste management from these early freshwater systems. Pictured here is famed Xenobiology 1 paleoartist Sean Ring's paintings of Saphia ziwa. Creature design by Sean and Source Blooded. Saphia ziwa have lost many of their ancestral Xenoeule segments, with only 4 remaining. The first segment is the head, consisting of a simple mouth, elongated flippers for mobility, external gills, and a pair of simple eyes. The following 3 segments repeat this process, though the flippers and external gills begin reducing in size and are positioned more posteriorly. These small limbs are used primarily for holding onto eggs after mating. Mobility is the primary use of the elongated fins on the head segment. Saphia ziwa are true hermaphrodites, who will gather to spawn several times a year, usually after rainfall. Members will perform a cloacal kiss, exchanging gametes and separating. They will do this with several partners to increase genetic diversity. After 2 local days, the Saphia ziwa will lay an average of 40 eggs that it will carry with its gills and fins until they hatch. Saphia ziwa emerge from their eggs on average 7 local days as miniature versions of the adults. They will reach adult size on average after 60 local days, and will be able to breed in an additional 30 days. Saphia ziwa are found exclusively in the freshwater ecosystems of Yama, preferring slow-moving water, as fast-moving rivers will often wash them away. Saphia ziwa are detrivores who feed on retinal phytes, decomposing material, and fecal matter of other organisms. The eyes along Saphia ziwa's dorsal side are small and simple, only being able to distinguish light from dark. Genetic ancestor, Xenoeule kirbii. Scientific name, Lacuslautus speculo. Origin, Xenosegmenta. Lifespan, 2 local years. Average length, 2 millimeters. Alright, buckle in because we have another long one. It's the sea sweeper. In the calm, tranquil waters of the great expanse, a cloud appears in the distance. In any time before this, such a cloud would certainly be a large school of plankton in a great, empty expanse. However, now, that is not the case. As the plume slowly moves below the surface of the ocean, a slight orange shimmer can be seen, as the true nature of the cloud begins to come into view. Tiny fish-like organisms swim in schools of hundreds along the upper layers of the pelagic zones of Almaish's deeper oceans, filter-feeding plankton from the clear waters of the ancient oceans. These organisms travel great distances over the planet, and can be seen anywhere the water is deep enough for them to avoid the coastal predators of the Yama-Kubshai reef system. While they may look like Misa, they are actually members of Xenosegmenta. They belong to a basal-clade of organisms that developed the eye and cartilaginous backbone early in their evolution, the cephaloptins. These particular cephaloptins have evolved a specialized mouth and digestive tract to efficiently filter feed in the deep water, far away from predators. They are the Catharians. The order Catharia appeared sometime around 20 million years ago, as esoterican cephaloptins were colonizing the pelagic zone. What triggered this move was likely heightened competition in the more competitive coastal ecosystems. Interestingly, there is fossil evidence that the first members of this order evolved within the coastal ecosystems, and only afterwards radiated outwards. If this is the case, as it is likely, then Catharia is mainly associated with the pelagic zone due to all coastal dwelling genera of the order having gone extinct by this point. One of the best representations of the basal Catharians are the members of genus Aquacomdenti, also called the sea sweepers. These organisms have global distributions and can be found anywhere the water is deep enough to protect them from predators. While their biology is fairly simple compared to their relatives, its efficiency has led them to becoming one of Almaish's most successful filter feeders. Pictured, a school of common sea sweepers is feeding during the early afternoon. Creature design by Lethal Cuteness. Like all other members of the clade, all members of Aquacomdenti possess a complex camera-type eye with the ability to see color. However, members of the Aquacomdenti possess two different cone cells that are susceptible to light at opposite ends of the visible spectrum. This ability is likely used to help them detect variations within the water, as many of them are preyed upon by larger animals that prowl the pelagic zone of the continental slopes. Aquacomdenti also shares a flexible cartilaginous bone that runs from the end of the tail to the base of the head with all other members of this clade. This bone, called the stem, is used to provide support for the muscular structure of the tail and lateral fins, and is critical to the anatomical organization of not just the genus, but the entire clade. While it is strong enough to provide a proper anchor for its tail and fin muscles, it is also quite flexible, resulting in Aquacomdenti being able to possess both great dexterity and strength compared to other creatures of similar size. Other cartilage formations exist within the fins, but this cartilage is much harder and is used to hold the shape of the fins firm as they push against the water. Along each lateral side of the stem, several bands of nerves run along the length of the organism. These nerves contain numerous small nerve clusters, one on each side of the stem in each segment. These nerve clusters have shrunk in comparison to their ancestors, with the exception of the three pairs, one in the foremost tagma, and one each at the end of the abdominal tagma. These pairs have actually begun to fuse, forming simple primitive brains. The core function of the nervous system are divided among them. The foremost brain primarily handles environmental stimulus, movement, behavior, and basic memory. The second regulates internal organs and most automatic functions, and the third controls reproductive functions and bowel movements. The foremost of these clusters is by far the largest, mainly due to it being responsible for movement and processing of visual input from the eyes, the most important sensory organ of this family. The circulatory system has also undergone significant improvements in this species. The central cavity has instead become a simple, yet large, network of passageways extending through the musculature of the organisms. These passageways are quite intricate, but not so complex as to require a proper heart, valve, or other specialized form of mechanism to keep blood pumping. Instead, simple muscle contractions during movement are sufficient, which works quite well considering Aquacomdenti are almost always moving to filter feed. While this system lacks the benefit of specialized fluids and a valve-sealed system, it is far more complex than the circulatory system of most more basal forms. As a result, Aquacomdenti is able to employ a faster metabolism and enjoy more efficient internal gas exchange without having to create more complex external structures. The external body plan of all cephaloptins, including Aquacomdenti, follows the same basic design: long, smooth bodies with a reduction in visible segmentation on the exterior of their bodies. This tagmatization has occurred, and members of the order possess three distinct tagma: the head, or foremost tagma, a central abdominal tagma, and a posterior tagma called the tail. The lateral swimming appendages of its ancestors have condensed down to two pairs, near the front of the abdominal tagma, and an additional smaller pair near the end of the tail tagma. The four appendages near the front of the body have increased greatly in size in comparison to more basal orders, and they have retained their ancestral cartilaginous internal plating, which provides structure and strength while swimming. These primitive fins are used to aid in steering. The smaller hind pair of fins are used in stabilizing the Aquacomdenti as it swims. Members of Aquacomdenti also possessed a vertical paddle at the end of their posterior tagma, and this served as the primary mechanism for propulsion in the water. In Aquacomdenti, these generalities manifest themselves in the form of long, ovular heads with gaping mouths and slender, smooth bodies. The head of Aquacomdenti is significantly larger than that of its relatives, giving the impression of a more compact body. But this is not the case. Rather, the Aquacomdenti's head has significantly expanded in order to capture and funnel as much water into their digestive tract as possible. While this has resulted in the organisms becoming a bit slower and less hydrodynamic, it has enabled them to become more energy efficient by increasing the amount of plankton that funnels into the stomach. Interestingly, most of the interior of the abdominal tagma is composed of a significantly expanded stomach. The stomach is located just behind the ciliable throat and is used to filter plankton from the large quantities of water it ingests. The water is funneled directly to the posterior anus where it is expelled. When the sea sweeper has filtered enough water, it will then seal its throat and flood its stomach with enzymes to digest the plankton. The digested nutrients are then funneled to the back of the stomach and filtered into the bloodstream, while the waste is sent down to the posterior anus to be expelled as the creature resumes filter feeding. Additionally, the first two pairs of gill fronds have hardened and no longer function as such. Rather, they are used as a means of laying eggs without having to return to the shallows, where predation is more common. All members of Aquacomdenti are diploid hermaphrodites, with each developing one male and one female gonad. Therefore, they will both lay eggs and fertilize eggs as a form of mating, and are theoretically capable of fertilizing their own eggs. Whether or not they do so is dependent on each individual species' instincts. Averamicia, also known as the common sea sweeper, these organisms rely on their schooling behavior to mate and are typical of their genus. Mating season begins at the end of spring and it ends in the middle of fall. This is relative to the part of the planet they are on. Populations near the equator mate year-round. All members of this species first lay their eggs by curling their posterior anus towards the base of their head, and then releasing up to 20 eggs into the large, non-functional gill fronds at the ends of their heads. The eggs are coated in a sticky mucosal membrane, which helps them stick to the gills. Then individuals will choose a nearby member of their school and proceed to fertilize each other's eggs. They do this by swimming next to each other and curling their tails together, positioning their posterior anus over the gill fronds. They then release their male gametes. Amacrica. The ridge sea sweeper employs a radically different reproductive strategy, more akin to that of organisms in the Yama-Kubshai reef systems, however reliant on the much safer Catharian ridge. The ridge sea sweeper schools will mix and intermingle during this time as they search the sediment for a suitable location, such as an outcropping of rocks or shallow hole. Once a suitable location is found, the school will lay all their eggs, approximately 20 per individual, into a large cluster. Then the entire school will proceed to fertilize the eggs. While the system does allow for a singular ridge sweeper to fertilize its own eggs, it also allows for the most diverse possible gene pool per egg, as any one of the mating individuals could fertilize any particular egg that has been laid. After the eggs have been fertilized, the school will leave the site and eggs to fend for themselves. A. Ditica. The western sea sweepers, like their close cousins the common sea sweepers, heavily rely on schooling to mate, and both lay their fertilized eggs in a similar fashion. However, where the western sea sweepers differ from their more cosmopolitan siblings is how they employ the geography of their habitats in the first and second phase of their life cycles. Mating season for the western sea sweeper occurs at the beginning of the local spring and ends in the final weeks of the local autumn. Unlike the common sea sweeper, this rule applies even to those populations living closest to the equator, as their range is almost entirely south of it. Roughly once every 30 days, the school will journey to the shallower parts of their range along the coast of Kubshai and the western ridge. Much like the common sea sweeper, members of the school will form pairs after laying their eggs, and proceed to fertilize the other partner's egg by coiling their tails over each other and ejecting the male gametes out of the posterior anus. However, there are two significant differences. First, the eggs are smaller and laid in far larger clutches. A single clutch of eggs can number into the hundreds. Secondly, the eggs are only allowed to develop on the fronds for a single day before they hatch and are released into the water. After this, the western sea sweeper will mate again, repeating the process until the school leaves the shallows. A. Australialis. The southern sea sweeper is a population that has branched off from the common sea sweeper in and around the south pole of Almatia as a response to low predation and highly seasonal climate. The mating season occurs near the end of spring, roughly around the same time that the sun rises over the horizon for the last time before the long Arctic day. This is triggered by a small portion of the brain made up of a vestigial remnant of their ancestors' other, more basal eyes. This photoreceptive part of the brain, called the neurocircadia, triggers the onset of mating season after a certain amount of time has passed without nighttime occurring. Interestingly, the mating season is incredibly short in the species, only lasting about 2 weeks. Like the common sea sweeper and western sea sweeper, the southern sea sweeper uses its ventral cranial gill fronds to house the eggs while they are fertilized and developed. The school will lay its eggs by coiling the tail and releasing the eggs through the posterior anus and will fertilize another sea sweeper's eggs through the same process, but with the additional process of crossing tails. However, while in most species the eggs hatch in a matter of days, it takes the southern sea sweeper a full 7 local days for its eggs to hatch. Furthermore, southern sea sweepers lay only 10 eggs, half the amount of the common sea sweeper. A. Veramicia. Eggs usually hatch within 5 days, with the naiads being bound to the safety of the fronds until they are large enough to swim on their own. The gill fronds are wide with numerous fibrous appendages similar to the baleen of Earth's whales. This allows the gill fronds to capture plankton as adults swim, providing food to the naiad. Furthermore, the naiads are supplemented with a small yolk that provides food for the first few days of its life. If there is a lack of plankton in the water, the naiads are not against eating their smaller siblings, who simply did not develop fast enough. This has resulted in larger litters when food is abundant and smaller litters when food is scarcer. This has also resulted in natural selection becoming stronger at weeding out runts in the event of a prolonged food shortage. After approximately 14 local days, after approximately 2 weeks, what remains of the naiads have developed enough to swim independently. While they are still much smaller than the adults and may struggle to keep up with the school, they are freed from the gill fronds. It is around this point that their mouths complete the move from the ventral side of the head to the anterior end, as in the adults, which is located at the front of the head, similar to Xenoeuli kirbii. Hence, they are able to filter feed independently. Roughly 5 days after this, their gonads finish maturation, and the juveniles become adults. If the mating season is still present, they will begin to lay their own eggs and fertilize others, which will in turn lead to more juveniles being born, and the school will continue to grow. A. macrica. Eggs hatch after just 1 to 2 local days, when small naiads emerge. These naiads are identical to the adults, except that they are much smaller and have their mouths located on the ventral side of the head, as opposed to the adult position. These naiads will first feed on plankton as they drift through the water, near the benthic layer of the ridge, until they are large enough to swim. The oral cavity does not move towards the adult position at all during this phase, and is generally regarded as phase 1 of the creature's juvenile life cycle. Some naiads who do not grow fast enough may drift out into the pelagic zone. These will be unable to feed algae to fuel their phase 2 metabolisms and will almost certainly die. Phase 2 of the juvenile state begins when the naiad is able to swim on its own. This phase usually begins after 2 to 3 local days. When this phase begins, the juveniles will swim to the benthic layer and begin to feed on algae and plankton organisms that live on the seafloor. It is during this phase that the oral cavity moves from the ventral side to the adult anterior-facing position. As the juveniles grow over the next 2 weeks, they will begin to exhibit the same schooling behavior seen in adults. However, schools at this stage are not quite yet able to intimidate smaller predators, and due to their dependence on the benthic layer, many juveniles will die to the few ambush predators that may stalk the edges of the ridge. Adult life is characterized by the completion of the oral cavity's migration across the lower side of the head, and the completion of the development of the porous membrane within the throats and stomachs to trap plankton. This process is usually completed around 14 local days after the second phase of life begins, upon which the schools will begin to break up and journey out into the pelagic zone surrounding the Catharian Ridge. Members of this species never stray far from this ridge due to their dependence on it to mate. The ridge sea sweeper is the only member of its genus that does not have a mating season of any form, and instead mates roughly every 30 to 60 days year-round. The gametes complete development after an additional 7 to 14 local days, after which the young adult is able to mate and will lay their own eggs when they return to the benthic layer of the ridge. Ditica. Once the naiads have hatched and left the fronds, they will feed on other plankton members of the ecosystem until they can grow large enough to swim freely. During this period, they are some of the most abundant food sources for the local fauna, and most naiads will be devoured during this time by larger organisms. After 10 local days, when this phase of the western sea sweeper life cycle ends, roughly 85% of all naiads hatched will be eaten. Hence, the amount of naiads hatched per school tends to be in the hundreds of thousands. The naiads will undergo rapid growth during this phase, and those that survive will be miniature adults by the time they reach the second phase of their lives. The major exception to this is that their fins will remain somewhat underdeveloped compared to their size, and the slow naiads will supplement their diets with algae until they are strong enough to withstand the open ocean. Once the naiads have attained this size, they will journey to the benthic layer to supplement their diet with algae to aid in their rapid growth. During this phase of life, they remain vulnerable to predators of the coastal ecosystems and competition with other algal feeders. However, they have 2 advantages. The first is that they are still capable of feeding on plankton, and the second is that this phase of life is relatively short, lasting only 3 days. In that time, the juvenile will eat almost continuously, desperate to finish their final stages of development so they can retreat to the relatively safer pelagic zone of the open ocean. In this phase, at least 1/4 of all juveniles who survive the previous phase in the life cycle will die due to being eaten by larger predators. Once the fins are large enough, the western sea sweeper will journey out into the pelagic zone to join a school. This is feasible for them due to the smaller range of the western sea sweeper compared to other members of this genus. When a school is found, the juveniles will join it and will likely spend the rest of their lives with that school. After an additional 3 days, the gametes finish development and the surviving sea sweepers become ready to lay eggs of their own. A. australialis. When the eggs hatch, they are effectively miniature adults, fully equipped with developed fins, eyes, and digestive tract. The young hatchlings even begin life with the ability to swim freely. However, they will generally remain latched to the nursery fronds until they reach 2 centimeters in length. During this phase, they will continuously filter feed, much like the adults would. Due to the lack of spending their energy free swimming, the young hatchlings are able to reach this size fairly quickly in approximately 5 local days. Since they are not really naiads upon hatching, this phase of life is simply referred to as the hatchling phase. Once a juvenile reaches about 2 centimeters in length, it will swim free of its parents as a juvenile member of the herd. Over the next 2 weeks, it will grow rapidly towards its adult size as the polar plankton is most productive during this time of the year. Within 5 local weeks, the youngling will reach its adult size. However, its gonads will not mature during this time, nor will they for the rest of the year. The gonads will only mature later into the next spring, around the beginning of the next mating season. This is mostly due to the long polar night's approach, as the resources spent on maturing the gonads would be better spent stocking fat reserves, as they are needed to survive the winter, while gametes will pose no usefulness until the mating season arrives afterwards. A. veramicia. The common sea sweeper has one of the largest ecological ranges up to this point in Almatia. The pelagic filter feeders can be found almost anywhere in Almatia, save for the majority of the continental reef systems and those areas where they have been outcompeted by their more specialized relatives. In their ranges, they are among the most common animals to be found. The common sea sweeper is, like all other members of its genus, a generalist filter feeder that is constantly filtering water through its body for plankton. However, they also lack the ability to pump water through their bodies and can only filter the water when they are moving. As a result, the schools of sea sweepers moving all across Almaitia's ancient oceans seem to never stop. and are almost constantly on the move. This is one of the primary reasons for their range being so large. Because the common sea sweeper is almost always moving, it and its school will traverse great expanses of open ocean during its lifetime, even if it does so at a calm and peaceful pace. A. macrica. The ridge sea sweeper is endemic to the Catharian Ridge and the surrounding oceans. While it is well adapted to the filtering plankton and could probably survive in a greater range, the schools of ridge sea sweepers remain close to the Catharian Ridge due to the need to return to it regularly to mate. This condensed range has resulted in the ridge sea sweepers beginning to specialize to the particular composition of plankton in the area, and they are more efficient at feeding there. These two factors have reinforced the ridge sea sweeper's established niche as a small generalist filter feeder, occasionally placing it in competition with the more common sea sweeper during times of surging birth rates. The ridge sea sweeper is better suited to this region than the common sea sweeper. This has led to the common sea sweeper being outcompeted in the northern regions of the ridge. A. ditica. The western sea sweeper lives in the warm oceans to the southwest of Kupchai. These regions include the western ridge and the southern portions of the Yama-Kupchai reef system. As a result, the western sea sweeper has adapted to the diet and pressures of its own region. It spends most of its time in the deeper pelagic zones, where predation and competition is lower. However, it journeys to the shallower areas of the ocean to spawn, allowing for a degree of niche partitioning between adult and juvenile populations. A. australialis. The southern sea sweeper has adapted to live in the Antarctic circle of Almaisha. While this region is not frigid, it does experience extreme seasonal changes due to the long polar night and day. A. verimicia, the common sea sweeper, is not known to possess any form of specializations regarding a specific type of plankton. Rather, their wide array of digestive enzymes are quite generalized, and they can feed on most varieties of plankton found in Almaisha's ecosystems. Interestingly, they struggle to feed on bloomers due to their lack of proper enzymes to digest them. Though they can get energy from them, it is much less efficient than with most other types of plankton. A. macrica, the ridge sea sweeper, due to its smaller size, has more specialized digestive enzymes to digest the plankton species surrounding the Catharian Ridge. As a result, they have gained greater efficiency in gaining energy from their ecosystems, which has helped it outcompete its relative, the common sea sweeper, in the northern part of its range. A. ditica. While most species of aquacomdenti struggle to feed on bloomers, the western sea sweeper has actually developed the proper enzymes designed specifically to digest them during its excursions to the more coastal waters. This has resulted in their ability to thrive in more coastal ecosystems, even though they prefer to stay in the safer pelagic layer of the open ocean. Other than this, their digestive enzymes have begun to specialize to the local planktonic populations, however, to a much lesser extent than other regional members of their genus. A. australialis. The southern sea sweeper's filter's digestive enzymes are more specialized than those of any other species within its genus. This is mainly due to its highly specialized life cycle within the Antarctic circle of Almaisha. The digestive enzymes are specialized to digest the plankton of the region as efficiently as possible, so that energy may be stored for the long polar winter. For this reason, the southern sea sweeper has the most efficient digestive system for its habitat out of the entire genus of Aquacomdenti. The southern sea sweeper also has much greater dietary needs during the winter and autumn during the year, largely due to a great restriction on its ability to feed in the long winter. As a result, the southern sea sweeper puts on a great deal of fat during these months and needs to consume additional plankton to accumulate this fat. A. australialis, the southern sea sweeper, is the only known member of Aquacomdenti that undergoes a yearly biological cycle other than a mating season. This cycle is timed around the long polar day and night, which dictate the productivity of the polar plankton, the food source of the southern sea sweeper. In early springtime, the first sunrise after several months triggers a response from the circadia, causing the metabolism to gradually increase from its winter state as the length of the day increases. The dim light that bathes the sides of the organisms is detected by its complex eyes and numerous internal circadians, vestigial remnants of the eyes its earliest ancestors had and lost due to evolution. These circadians and the complex eye send signals to the neurocircadia, which is what triggers the end of hibernation. As the amount of daylight increases, the southern sea sweeper's bodies gradually stir to life. As the spring goes on and the plankton become more productive, the sea sweeper's metabolism will continue to rise, keeping pace with the change in season until about late spring when it reaches its summer levels. Once the daytime becomes significantly longer than the night, signaling the coming of the long polar day, the southern sea sweeper's bodies prepare to mate. In the young adults, the gonads finally complete development, and in older adults, the production of healthy male and female gametes resumes. The mating season will begin here, and it will last for a short 2 local weeks. After this, the gonads will cease production of gametes, and focus will shift to stocking up on fat stores for the winter and population exchanges with other schools to maintain genetic diversity. As the long polar day draws to a close, the neurocircadia begins triggering a gradual slowdown in the sea sweeper's metabolism. This decline will continue all through the autumn until the beginning of the long polar night. The signals to begin this slow shutdown are triggered by the first nightfalls detected by the circadians. During this phase, the southern sea sweeper's body begins to swell with stored food and an early version of fat cells. These will be used to help the sea sweeper survive the long polar night and hibernation. When the sun sets for the final time and the long polar night begins, the southern sea sweeper's metabolism nearly slows to a halt. The cold, combined with the circadian system, forces the sea sweepers into an almost catatonic state. They are still conscious in a loose sense of the word. Their bodies are active and they maintain a loose sense of awareness of their surroundings. However, their state is most comparable to that of sleep. During this phase, the sea sweeper will not eat and will barely move, doing everything it can to conserve its winter storage of fat and undigested plankton until the spring. The genus Aquacomdenti has schooling behavior. Individuals are inclined to gather with and follow other members of their species. This can result in major population exchanges when two large schools encounter each other, sometimes resulting in a merging of the schools. Aquacomdenti's eyes are always placed in dual pairs near the anterior end of the head, one pair on each side. These pairs have moved away from the median, yet these organisms also demonstrate depth perception. They perform this ability by employing stereopsis with each pair of eyes. This allows them to perceive depth on both sides. However, it has created a larger blind spot near the median line. Performing stereopsis twice is quite mentally taxing, and most of the brain is now devoted to processing visual data. The optical portion of the brain has swelled and divided into a left and right section. Each of these sections is responsible for processing information from the eye pair closest to it. These sections are connected by a single neuronal structure called the optical median corpus, which is used to share information between each portion of the optical brain. If this nerve cluster were to be severed, the individual would not go blind. Each side has its own connection to the rest of the brain. However, such an event will be catastrophic for its ability to process visual data coming from in front of the organism, as the lack of communication between these two brains prevents visual analysis of that region. Interestingly, Aquacomdenti veramicia, the common sea sweeper, shares its environment with another larger filter feeder, the night sailor. The escort juveniles do not attack the sea sweepers, as the sea sweepers are simply too small to agitate them. As a result, common sea sweepers will swim alongside grazers if they see them, enjoying the protection of the escorts to filter feed in safety. The grazers also gain from the presence of the sea sweepers. Due to their poor eyesight, they struggle to see predators approaching. However, sea sweepers have highly advanced eyes and can detect these features. Thus, grazers can detect the behavior of the sea sweepers and use it to gauge the presence of nearby predators. Genetic ancestor? Xenoeuli kirbii. Scientific name? Aquacomdenti species. Origin? Xenosegmenta. Lifespan? A. veramicia, 3 local years. A. macrica, 7 local years. A. ditica, 3 local years. A. australialis, 12 local years. Average height? A. veramissa, 1 cm. A. macrica, 3 cm. A. ditica, 1.8 cm. A. australialis, 5 cm. Average length? Veramicia, 5 cm. Macrica, 15 cm. Ditica, 9.2 cm. Australialis, 20 cm. Okay, well, if you're still with me, we have 2 more organisms to go. They're shorter, though. The first one is Umtrabella. With the development of coral-like organisms who shape their environment like the jade crowns, some species of chloroseudopods develop to exploit the fruits of other species' labors. Umtrabella is one such development. Adapted to cling to the sides of coral shelves, and more often than not the side of jade crown colonies, this chloroseuropod descendant supplements its photosynthetic diet with plankton to a much greater degree than its ancestor, feeding on the microorganisms even during the day by exploiting the currents running off the shelf of the jade crown colonies. Pictured here, a pair of Umtrabella absorb sunlight while their tentacles wave around in hopes of snaring small organisms for extra nutrients. Creature designed by Dr. Misa and source-blooded. Physically, Umtrabella is rather similar to its ancestor, possessing a singular eye spot that is sensitive to light and movement, 2 tentacles capable of filter-feeding organisms, and a long trunk with 5 basal feet. The major change besides the overall larger body size is the positioning of the shape of the umbrella, the photosynthetic hood. Where in the chloroseuropod it was positioned radially around the eye spot and feeding tentacles, Umtrabella has repositioned it into a dorsal frill that can be used as both a photosynthetic organ and a net to aid in the capture of plankton. The walking feet are capable of secreting a sticky substance or lubricating substance, depending on the orientation of the organism. It will secrete a sticky substance when the organism is climbing up, and the lubricating substance when it is traversing across the ground, usually to mate, a time of high mortality. Umtrabella are hermaphrodites who will gather once every other full moon to release gametes into the water. Umtrabella start life as a simple, worm-like photosynthetic organism barely 3 centimeters long. During this time, they are fully photosynthetic and living on rocks or retinophytes. After a period of 2 local years, they have grown to about 10 centimeters in height and have developed their umbrella and walking feet. At this stage, it will begin incorporating plankton into its diet and move to living on coral shelves, preferably jade crowns. At 3 local years, they become sexually mature and will participate in releasing their gametes. Umtrabella can be found in the coral reef systems between Yama and Kupchai. They also have a preference for living on the side of jade crown colonies. Umtrabella are photosynthetic organisms that supplement their diet by feeding on microorganisms like plankton. In addition to the central eye, the webbing of the umbrella on Umtrabella is sensitive to touch, so as to help its feeding tentacles find prey items who have become trapped. Genetic ancestor: Chloroceutopod. Scientific name: Umtrabella rupiaceo. Lifespan: 5 local years. Average height: 30 centimeters. Alright, and last but certainly not least, we have Vesicapiscus. With the competition in the oceans growing larger and larger, many species have moved into the freshwater biomes. Though the rivers and lakes of Yama are no less dangerous, direct competition for food is more rare. Many of Yama's lakes are now dominated by slow-swimming yet armored schools of Vesicapiscus. Vesicapiscus are descendants of Durospina, who exploited the lack of free-swimming competition and became free-swimming themselves. Feeding on an almost exclusive diet of Leucostopedes leaves and stems, they are the gardeners of Yama, keeping the retinophytes from overpopulation and clogging waterways. Pictured here, a Leucostopede is foraging near the shore of a lake. Creature designed by Saurus-blooded and Kipzilla. Vesicapiscus have a compact body that is covered in a thick keratinized dorsal shell, which is also present on the cranium. A dorsal spine has formed partially for defense against predators, but primarily to stabilize the Vesicapiscus swimming. The cranium has extended to house larger eyes and antennae, so that Vesicapiscus can better identify members of its school and threats in the murky waters of lakes and swamps. The mouth has also extended, creating a distinct upper and lower jaw. The upper jaw is part of the keratinized dorsal shell and is covered in sharp ventral grooves used to masticate retinophyte material as it moves down the mouth, while the soft lower jaw will match the retinophyte material against the grooves to accelerate the process. This is to allow for easier digestion of the relatively nutritionally poor Leucostopede leaves. As with the ancestral Durospina, the pair of claws on the lower lateral sides of the cranium are used for grasping food and deterring predators. The ventral pseudopods have undergone the most change, with the secondary pair going over the most extreme change. These limbs have fused to the ventral side of the body and have been converted into a bladder containing metabolic gases to control buoyancy. The first, third, and fourth pair of pseudopods have flattened into short circular fins that provide propulsion and stability when swimming. These limbs are still articulate enough to allow Vesicapiscus to walk along the bottom of lakes and rivers should the need arise. Vesicapiscus are true hermaphrodites, and a pair will usually dig a pair of holes in soft sediment with their hind limbs and lay their eggs, one in each hole, before fertilizing them and burying them. Vesicapiscus emerge from the eggs as miniature versions of the adults, but lack the dorsal shell that is formed of dead keratinized cells. Their development is accelerated during this stage of their life, and they will feed by means of filtering out sediment. During this time, they will develop their shells and be able to join a school of Vesicapiscus. This process will take just under a local year. After this, their metabolism slows down as their diet changes to retinophyte leaves. When a Vesicapiscus reaches the age of 4 local years, it will be large enough to breed. Vesicapiscus are native to the swamps, lakes, and rivers of Yama. Vesicapiscus start life filter-feeding sediment, but quickly shift to a purely retinophyte diet once their shell is fully developed. Vesicapiscus live in schools of, on average, 20 to 30 individuals. These schools are leaderless, and purely for protection from predators, they will often divide and reform multiple times during a year. Genetic ancestor: Durospina. Scientific name: Vesicapiscus loricatus. Origin: Xenotagmata. Lifespan: 8 local years. Average height: 10 centimeters. Average length: 30 centimeters. Alright, well that's it for this episode. I hope you enjoyed it. I have enjoyed making quite a few of these creatures, and remember, the World Anvil page for the project has more information, including information about the humans who study Al-Maisha. Just another reminder, if you enjoy this video, please hit the like button. If you disliked it, give it a thumbs down and tell me how I can do better the next time in the comments. Either way, please subscribe and hit the bell icon and turn on all notifications so you're always notified when there's new Dapper Dino content. Until next time, I'm the Dapper Dinosaur. I just want to take a moment to thank my patrons and channel members, especially those pledging $20 or above: Bob Knob, Bent Hovend, Ian Chen, Chris Love, Sphincter of Doom, The Venerable Bede, and Patrick Dennis. My patrons and channel members really help make this channel possible, and without them, this channel wouldn't exist. If you would like to support me, you can click the join link on this channel below this video, or you can click on my Patreon, link in the description, and pledge. On Patreon, there is an option for an annual subscription, which comes with a 10% discount. And joining the channel will give you access to my custom emojis. However, if a regular pledge isn't for you, I do have a merch store, And if monetarily helping out the channel isn't a thing you can do, every like, share, and comment really helps the channel.